So, gene therapy, the basics. Gene therapy has already been used to cure some genetic diseases, such as severe combined immunodeficiency syndrome. But the hope is that it could also be used to cure other diseases like cystic fibrosis. If we looked inside our bodies, we'd see that our bodies are made up of small, tiny cells like this. Genetic conditions occur when people inherit a version of a gene that doesn't work as it usually would, and this can cause problems for the cells. And when the cells don't work properly, that can cause a disease. An important part of gene therapy is being able to get a therapeutic gene, so a gene that's going to help cure somebody who's got a genetic disease, to the right part of the body, the part that's affected. So in the case of cystic fibrosis, we need to get it to the lungs, these two big organs on either side of the body here. So that sounds quite simple in principle. We need to get our bit of DNA into the cells of the lungs. So how do we do that? Well, we need to use a delivery vehicle, and that's something called a vector. So vectors take our therapeutic DNA from point A and they put it into point B, in this case, the lungs. We've got two different delivery vehicles, vectors, that we could use. We can either use a virus, like this, or we can use a liposome, like this. In cystic fibrosis treatment, most research so far has focused on using liposomes. To get the DNA into the body, we can either inject it, or we could use a nebulizer. Now, for cystic fibrosis, it makes sense to use a nebulizer because we're trying to reach the lungs. So a person affected with cystic fibrosis can inhale the liposomes using a nebulizer and get them into the lungs that way. So here's our liposome with the therapeutic DNA inside, and once it's been inhaled into the lungs, it mixes with the cells within the lungs. So here we have a lung cell, and on the outside is a therapeutic bit of DNA surrounded by a liposome. Now the cell membrane of the lung is fatty, like the liposome, and so the liposome and the cell membrane mix, the therapeutic DNA comes into the cell. So here we have our bit of therapeutic DNA. We've removed the liposome, and the therapeutic DNA crosses the cell membrane, comes through the cytoplasm until it reaches the nucleus. Once it's in the nucleus is like the command center of the cell, and it's where all the rest of the DNA sits and tells the cell how to work. Once the therapeutic DNA is in there, the hope is that it would be used by the cell so that the cell could work as it normally would do. So it doesn't actually matter if we don't reach or cure every single cell that is affected by a genetic condition. So long as we reach enough, then we can help cure somebody who has a genetic condition like cystic fibrosis.